this is the visual part, and now I'll show the visual hierarchy by itself. Here is the LGN, the visual cortex, visual V1, V2, V4 is here, and then the inferotemporal cortex up to the highest one for hippocampus up here. So they all end up there in the frontal limbic cortex. Here is this is the ventral stream for object recognition. Here's the dorsal stream. This kind of diagram is almost impenetrable. It doesn't help much. What you have to know is here each area is simply one little box. That box consists of millions of neurons. And each one of these lines is just a single line for each connection to any other area in cortex. So it's a very, very, very simplified diagram of what's actually going on. Here's another way of showing the same thing. Here, Lenny, in 1998, scaled the areas by how big they actually are in the brain. And you see V1 and V2 are huge. And when you go up the object recognition hierarchy, they get smaller and smaller. And same thing when you go up to the action stream, the dorsal stream, they get smaller and smaller. And here he has the density of connections is portrayed by the thickness of the line. So here you see that V2 has a massive projection to V4. V1 also bypasses V2 with a massive projection to V4. Even that is highly simplified, because each one of those bundles is millions and millions of fibers, and they connect these areas in a topographic point-to-point fashion that preserves neighborhood relations. Very important for what these areas do, preserving the neighborhood relations. Okay, so here, to try to show what this actually amounts to, what this connective pattern, what this actually amounts to, we're going to take the whole brain, and we're going to, oops, going the wrong way, take the whole brain. Here's a piece of the hierarchy. Here's one of those 3 boxes, and here are the lines connecting them. Now we're going to take those 3 areas, and we're going to pull them out of the system. And here is one area, 2 areas, area 2, area 3. Each one of these, as I said, is connected in a topographic fashion, in coherent fashion, to its neighbors. So this one, this part connects to this part, this part to this part. Same for this one. If we now take, because these are monosynaptic connections, fiber transmission is fast, so we collapse, we now shrink the fibers so that we put these areas by their topographic correspondence, and you get a stack of pancakes. These pancakes are in topographic register. They are in retinotopic register. Okay? It is not true what everyone says, and Rizeki has been preaching from the very first of his forays into V4 and the color space. It is not true that the brain takes things apart so that there is a binding problem. The brain always keeps things together in the topographic register to the retina, and that doesn't cease in the visual system until you head for the hippocampus, which has sparse coding. So all the way up the hierarchy, the topography gets coarser and coarser because the areas get smaller and smaller, but you always know which place color is in and so on. And this was solved by, it was pointed out by Kawato. This is simply for those who has heard about the binding problem. Lots of people have heard about the binding problem. Well, Kawato in a 1997 paper showed that it doesn't exist because of the topographic register. And I'll try to quickly go through it. Here's the eye, here's V1, here's V2, and here are 3 of the areas and 6 of the areas in the hierarchy. This is the object recognition stream, this is the action stream, the dorsal stream. And he shows the eye sees a red ball moving to the left and a blue cube moving to the right. So red and movement and ballhood is kept together in topographic registry, and what do you call it? Box hood, blueness and movement direction is because they are local in the topography. They are kept together, and on these higher maps, even on the highest maps, MST, AIP, and inferotemporal cortex, they are separate in the topography. There is never a mix-up between the blueness of the ball and the blueness of the box and the redness of the. So here he shows how it works. Here is primary visual cortex. If leftward motion, ballness, and circle hood, red circle hood, or blueness, blue squareness, cubeness, and rightward motion are always always projected back to primary visual cortex by so-called feedback projections. So everything is in register at all times. So we can forget about the binding problem. Every solution to it is a pseudo-solution because there isn't any problem in the first place. And you really have to read Kawato's paper to appreciate the elegance of his solution. So back to this. We have now collapsed these three areas into the pancake with corresponding registry. Now we collapse all areas in cortex. The somatosensory areas, the visual areas, and back here is a little bit, you can see a glimpse of the auditory area stacked up. You stack them up and you get this pyramid because as we saw in the Lenny diagram, the areas get smaller and smaller as you go up. I have made a separate stream for the object recognition and the dorsal stream here. I have separated them to a little tunnel here. So you go up. At every step of this hierarchy, it is a bona fide hierarchy because they are all connected, included not only with feed-forward connections, but with feedback connections. Each layer projects back to the previous layer, and often some layers further down the hierarchy. Because of this double projection, upward and downward, all of this is bound together into a single, I call it epistemic space. A huge potential space of representation that is an arena for doing the inductive inference and the abstraction operations and so on that are required to figure out what's out there. Here is the feed forward. Here I have put the layers. Here is one. Here is a cross section of one area, another area, another area. Here are many areas, K, L, M, N, O, P, Q, R. And here are the feed-forward stream of connections, feedback stream, and they are matched point to point in the topography. It's a powerful system, and here I have done it like a cartoon toy representation. At the top of the hierarchy sits the hippocampus. At the highest point of the hierarchy sits the hippocampus as a hinge. It's just amazing. It does exactly what it should at that position. It converts the supragranularly dominated feed-forward current to the infragranularly dominated feedback current at the hierarchical apex of the cortical system. If you can see here, and this is the feed-forward stream. It goes into the hippocampus and goes CA 321 subiculum, and it comes out as the feedback pathway to entorhinal cortex, parahippocampal, and neocortex. So feedback up and feed forward up and feedback here. Now every one of these levels also sends feedback in the same way. So we could do it without the hippocampus, but because the hippocampus sits at the top of the system, it has a unique functional significance, which we see when we lose it, like in HM. We're not storing memory anterogradely efficiently any longer. So, I will skip this for the moment. So here I have made a cartoon of this. The red feed forward goes all the way up, all the time, through all the areas, and all the time the blue feedback pathway is feeding things back, and they are working simultaneously at all times. Feedback and feed forward, they are going like this. It's called a countercurrent, and there are uses of countercurrents that are unique in biology.
But Bjorn, detail, but
yeah?
if you now link the hippocampus, that sits in the temporal lobe.
Yeah.
Right, so that stream has to be converging and diverging, and so how should I imagine that in this diagram?
By the connections, the young diagram is the easiest. It doesn't matter where they sit in space. Here, wait a minute.
No, look, I don't want to distract you, okay?
No.
Just to, because now the arrows go from occipital to frontal.
Yeah.
While
Yeah.
The hippocampal hinge is of course the temporal.
That's just,
structure.
yeah, here they are. I just drew an arrow one way through cortex, but of course, the fact there are very long connections to the hippocampus from some of these. So the frontal hippocampal connections are very long. That's why people haven't paid enough attention to them. When I saw the young diagram, I said, "Oh my God, the hippocampus is sitting by long connections as the spider in a web that I never thought of it, that it was connectively, if you disregard fiber length, it's absolutely sitting in the temporal lobe, inside the temporal lobe. So it's very long fiber connections to go up to the frontal, and they are there in huge, massive bundles. So this, to me, it was an eye-opener, that connectively, the hippocampus is sitting in the frontal limbic, smack in the middle of it. So this is, of course, just a toy cartoon. It goes here, it goes everywhere. These are long connections. What then is such an arrangement good for? It has many uses, but a hint at what this elaborate hierarchical cortical apparatus basically evolved for is provided by that which it does most effortlessly, routinely, and best. What is that? For example, allowing us to recognize the greater part of thousands of pictures after mere passive exposure for a few seconds. Here's the data, Shepard 1967. 600, imagine you are sitting in a chair in a psychology lab, and in front of you is a screen, and the experimenter shows you 612 pictures in a row for 6 seconds each. And it's 6 seconds, next image, next image, next image, next image. It takes a long time. The same day, several hours later, he shows you a scrambled version of a scrambled sequence of the same images, and he asks you, have you seen this before or not? And you answer yes or no. Now, out of 612 pictures, you have 98% correct. If a few years later, Standing showed 2,560 pictures to people for 5 or 10 seconds per image, 90% correct after 5 days. All right, this is a challenge to Louise. There is no time for consolidation between these pictures. Well, there is. They are being consolidated, absolutely. But this is a very different setup from showing something and having a delay period and then testing. Here is going one after another, bang, 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 bang. And of course, when we're moving out in the world, particularly if we're on a motorcycle, there are scene changes every second. So, and now the game was on. How far can we go with this? Standing's latest in 73 was 10,000 ordinary pictures. It took a day and a half to show them. 10,000 ordinary pictures, 5 seconds each, 6,600 correct after 2 days. That's quite a feat, okay? Aude Oliva at MIT is following up on this work today with modern methods, and she's having some very nice results. It provides you, in other words, with recognition memory, the capacity to tell whether you have been there before, seen that before, met so-and-so before, or done that before. Very, very useful things, and to know it very accurately. Which in turn means that every instance of experienced reality, every now, is framed by your prior history of experience, putting the powers of inductive inference at the heart of the cortical system. This is where prediction comes in. Inductive inference is the only process known to us by which essentially new knowledge comes into the world. This is Roland Fisher, who did his great contribution to population genetics, and all of our psychological standard statistical methods are from Roland Fisher, derived from his population genetics statistical studies. Roland Fisher, a genius, published mostly in the 1930s. So here is a crude version of mapping predictive inference onto the cortical hierarchy. The feedback stream, I have kept the same colors. It was blue before, it's blue now. The feedback stream feeds down predictions from a high-level model to a lower level. That level in its turn feeds down a prediction from that level to a lower level till you are at the prediction for the sensory primary information. The sensory primary information in its turn feeds up to the next level, induces changes the weights. If there is a discrepancy between prediction and observation, the error signal goes upstairs to the next level. You are changing the weights of your learning algorithm, and this goes, so all the time it's being fed up the hierarchy and down the hierarchy, and wherever there is an error, you transmit up to the higher level. And you hope your best that at some level the error is going to disappear, that you have high enough a model of the reality that you're confronted to make the error disappear. So again, here I have that ascending hierarchy and the descending context hierarchy, and you feed down the hierarchy to your predictions and your observations up the hierarchy, or rather your error signals up the hierarchy, and correct your weights and hope that things will even out in the end. Direction of a statistical movement pattern down there. It should not report this place or this place or this place. Report only on what once had a red circle around it. The red circle disappears and these four movement patterns show up. Here the movement statistics are down to the right, here they are up to the left, here they are random, here they are random. Now, this task is obviously a top-down. You are priming attention by the search is predicated on a top-down instruction. Report only the stuff that happens in the lower left quadrant. That's the movement you should tell me, and you tell it to me by moving your eyes from the fixation point in the same direction as you detect the movement you detected here. It's a very, very, very well-controlled study, and now the point is, everyone, including myself, before I saw this study, I would have assumed that it's obvious because the cortex has feedback pathways. The instructions are obviously up here in frontal cortex. This is the dorsolateral frontal things about well-trained, learned. The instructions, look where the red circle was before we showed the movement stimuli, is up here. It's a higher level instruction which the monkey has taken 2 months to learn, but it now performs at about 80% criterion, and you would think that the cortex instructs the lower level perceptual areas by feedback from the top and down. That's what I would have assumed. That's what everybody assumes, and I assume that you too, if you listen to me closely about the feedback connections, would assume the same thing. Lovejoy and Krauslis, they are physiologists, behavioral physiologists who are used with trained monkeys. They put to test what that behavior is dependent on. They put an injection cannula in the brain of the monkey they had trained, and they injected muscimol. Muscimol is an inhibitory agonist. It essentially silences the tissue where you injected it because all the inhibitory cells are going crazy and clamping down activity. So you're shutting off the area where you have injected muscimol. So the question is, here are the results. Before the injection, 0 to the wrong direction, 88% in the right direction, a couple of stray mistaken trials, 88% correct. The monkey indicated, here's the fixation point in the middle, and the movement was down to the left, this is the movement, and it indicated by its eye movement that it had detected the correct movement direction in the correct place. Here was the wrong place, 6% were wrong. Then they injected their muscimol, and after the injection, here's the fixation point, performance was completely deteriorated. 89% to the wrong place. Okay? Where was the injection site? Before I read this paper, I would have assumed the injection site was someplace in cortex that interrupted the feedback from high frontal areas to the sensory areas. That's where I would have put my injection electrode. But Lovejoy and Krauslitz, they are collicular people. They had their injection cannula in the colliculus. It was down in the midbrain, and it completely devastated performance on something that everybody assumes is a top-down presetting of sensory areas for the search image, okay, the search instruction. And it had nothing to do with the fact that it was an eye, you think it's colliculus, it's because eye movements. The eye movements were intact. When it moved the wrong way, they had no problem with eye movements, and they did the same task with reaching, with a button press. No eye movements involved at all, just fixation and a button press. The monkeys were just as devastated with the button press, so it's not specific to the eye movement thing. So this is a very strange finding. Here is the brain again. This is not the monkey brain, but the mouse brain, but we'll put the Lovejoy and Krauslitz monkey results on the mouse brain. Here were the instructions up in frontal cortex. When frontal cortex, in this task, not in every task, but in this task, when it wants to preset the sensory areas for the search image, it sends the instruction down to the colliculus. The colliculus sends instructions up to the pulvinar, and the pulvinar informs the rest of the cortex. Instead of going bang from top down on the feedback pathway, it goes this roundabout way through half the brain, down to the midbrain, and back up to cortex. So here is a clear example of an obligatory functional division of labor between two very different parts of the brain, the midbrain and neocortex, in what at first blush might appear to be a typically cortical top-down task. But the top in this case was via the colliculus. It was via the bottom, okay? And not the bottom of the cortical visual system. That was the third instance when he got up there. So from the topmost position down to the midbrain and back up to cortex. It looks like a roundabout, but fiber connections are very fast. The colliculus is as close to the frontal cortex as one of the cortical layers is from another cortical layer. Okay? One synapse to the colliculus, that's very fast. If it had gone on the feedback pathway through cortex, it might have had to pass 5, 6 synapses. We don't know. There are some shortcuts, but we don't know. And of course, there are others where cortex is dependent on other structures. By the 90s, people were starting to draw diagrams like this. Cortex in the middle, cerebral cortex in the middle. It's different areas. This is M1, this is motor cortex area B, C, D. Basal ganglia flanking it on one side, and the cerebellum flanking cortex on the other side, with cross connections. So apparently cortex is dependent on these other structures, and we have known for a long time all of those three structures are very big structures in the brain. The cerebral cortex is the biggest. Not the one with the most neurons. The cerebellum has the most neurons because of the small granule cells, which are millions and billions of them. But cortex is big, volume-wise and surface-wise. Cortex is the biggest. Cerebellum follows it, and the basal ganglia are also a massive collection of nuclei just below the cortex in the subcortical telencephalon. And they correlate with one another very well. If you look across mammals, in mammals with big cortex have a big cerebellum and big basal ganglia. In mammals with small cortex, they have small basal ganglia and small cerebellum. And here the correlation is with the brain as a whole, but because cortex accounts for most of the brain as a whole volume, therefore, here you see cortex accounts for a disproportionate amount of brain volume. As cortex grows exponentially, the rest of them follow very slowly. So, and if you now take volume of the cerebellum and you regress it against the volume of neocortex, you get a very sharp, nice correlation. So they are correlated, and the same thing goes for basal ganglia. They grow in tandem. So a big cortex requires a big cerebellum and a big basal ganglia. Here you have the maps. This is cerebellar, this is cortical output to the cerebellum, and midline view and lateral view of the brain in the monkey. And the black labels are areas that receive cerebellar projections. So a huge expanse of cortex, not just motor cortex. Everybody thinks that motor cortex is served by the cerebellum. A huge expanse of cortex is served by the cerebellum, and is projected to by the cerebellum gets projections from a huge expanse of neocortex. Same for basal ganglia. Here you have the striatum, caudate and putamen, the main components, the big voluminous components of the basal ganglia. Seen from above, you're looking at the brain from above, and here is the head of the caudate, and here's the tail of the caudate, and the pulvinar is packed in beneath here. And these are the territories of the striatum that receive posterior parietal input, orbitofrontal in the middle, and orbitofrontal anterior cingulate and superior temporal at the side here. So huge expanses of cortex project to the basal ganglia, and here are the let's see, what is this picture showing? These are the areas of cerebral cortex that receive return projections from the striatum via the thalamus, so-called cortico basal ganglia thalamocortical loops. So a huge expanse of cortex is also receiving from the basal ganglia. It's not just motor cortex. Here you have a summary of these things. Large expanses projecting to the cerebellum, receiving from the cerebellum via the thalamus, and the cerebellum is also directly connected to the basal ganglia. And here, corticobasal ganglia projections. Given the anatomy of this triple tandem relation, what about the functional division of labor? What are these structures contributing to their ménage à trois? Cerebellum, basal ganglia, and neocortex. What is cerebellum and basal ganglia contributing to the cortical labors? What are the basal ganglia for? What is the cerebellum for? What is its generic function that it might contribute? Because it contributes to so many other places besides cerebral cortex. Same for basal ganglia. So here are some guesses in the literature. This is from Hauk from a while back. Now, if you had to guess to guess reward propensity for the basal ganglia is it's pretty minimal. You know, if you're going to have any guess, I mean, to guess reward propensity is not a tall leap of faith. Okay, if there is one thing that we have heard to death about, it is about the reward system and the dopamine system in the substantia nigra, pars compacta, and the striatum. So here is his guess. Pattern classification and embodiment are also not very heavy-duty guesses about what the basal ganglia are. Same for cerebellum. An error signal, refinement and amplification. You mean that cortex couldn't amplify its signals if it wanted to? It can't be that. And besides which, cortex itself, as we saw in the previous slides, is heavily involved with its own error correction procedures. So it's not error correction as such that the cerebellum is contributing, but these are guesses. Here's another guess by somebody else. Here we have cortex both above and below, output from cortex and input to cortex from basal ganglia and cerebellum. And here the guess is that the basal ganglia contribute to cortex suprasecond timing, and the cerebellum contributes to cortex subsecond timing. Well, it's a guess, you know, cerebellum is capable of quite some fast things, but the basal ganglia, as Tony Prescott knows, has, for example, the novelty spike, which is a fast signal, bang up to the basal ganglia. And that's subsecond. That's certainly a couple hundred milliseconds, less than that maybe. So
it's very fast, too fast to go by.
Yeah. So here you have a fast signal, so it can't be suprasecond timing. The basal ganglia cannot be relegated to slow processes. It is both fast and slow, and the cerebellum, of course, has to do with slow signals because the hypothalamus has a loop through the cerebellum. So the cerebellum isn't fast as such, in some cases it's a fast signal. When you feed a fast signal in, it's going to handle it fast if it has learned the appropriate prior, so to speak. So this is also not a very convincing thing. Here is how a few years later, incorporating some ideas from Doya in his scheme, you still have cortex in the middle, basal ganglia on the left, and cerebellum on the right. Now he revised it from error learning to supervised learning. This is a bit better. He called cortex Hebbian learning, that is unsupervised learning, and he called the basal ganglia still reinforcement learning. Well, let's go to the horse's mouth for that idea that Hauck took up. It's Doya in the year 2000. He had a very elegant division of labor between neocortex, basal ganglia, and cerebellum. And that was neocortex, unsupervised learning, basal ganglia, reinforcement learning, and cerebellum, supervised learning. Wonderful. I think that is on the right track. Okay, they are all big structures, and big structures store information. So it's gonna be some kind of learning function that is the generic function for each one of these, because each one of them is big. It's sort of like a storage capacity. And here is the first step towards actually getting a handle on a sensible division of labor. But of course, unsupervised learning for cortex, reinforcement learning. Obviously, there are things that go through the amygdala and cortex which are reinforced. Reinforcement is not going to stay confined to basal ganglia and being shipped out. Reinforcement is much more than the dopamine signal. When you have a complex contingencies and so on, you have all kinds of things going on directly into cortex that doesn't first stop in the basal ganglia. Supervised learning for error, as we saw, error correction takes place in cortex. So supervised learning of what? We still are looking for a more precise definition of a generic definition of the division of labor between basal ganglia, cortex, and cerebellum. I can't give you the final answer, but what are the guesses for what this supervised, what the reinforcement learning is affecting in the basal ganglia? The basal ganglia are associated with things like action selection, with things like sequential behavior, with things like serial order in behavior. And my guess is that the generic function is going to be, for basal ganglia, is going to be in this something to do with timing and sequencing and serial order, okay? And that the reinforcement learning goes in to cement, to fix some of the parameters under which these action selection and so on are done. For the cerebellum, the supervised learning of the cerebellum requires an error signal. Now, most people are dealing with cerebellum in motor terms. They have a motor error signal, but as Paul Dean has pointed out, there is no motor error signal directly available in the brain. Most of the time, we see that we are in error by our motor, for example, a reaching movement, when we miss the target. Now, missing the target isn't derivable directly from a motor error signal. It's a sensory mismatch signal. The target is here, my hand is there, I missed the target. This is a sensory signal, a visual signal. Yes?
Also, if we follow your hypothesis that the cortex is performing this reconstruction,
Yeah.
The reconstruction itself would require also the computation of some kind of error.
Absolutely.
Whether just for learning.
Exactly. And you
so
saw
it would kind of be supervised or something.
the cerebellum is doing something. As you saw in my previous diagrams, there were errors in the cortical hierarchy, error, all up and down. So error is not enough. We have to know more specific. And one bid for that is Paul Dean, who has proposed that the general function of the cerebellum is decorrelation. And when you decorrelate the sensory error signals, you can get the motor error sort of for free. And I won't go into details about that. Here is Paul Dean and Stone, their proposal for cerebellar decorrelation. This is modeled for the VOR the cerebellum takes the 6 eye movement commands as input and supplies corrections to the 3 vestibular signals based on sensory mismatch, based on sensory error. Okay? And this VOR, of course, is the vestibular ocular reflex that resets the eyes when the head turns. When you turn the head, the eyes go tick, 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 resetting to cancel retinal motion as your head moves. So, at least a modicum of assurance about the division of labor between these 3 major learning capacities of the mammalian. Given at least a modicum of assurance about the division of labor between these 3 major learning capacities of the mammalian brain, we come to the final clause of the title of my tutorial. How do they, or any neural systems, work together? That's the holy grail. Paul is working on that. Everybody is, in a sense, working on that. How do they work together? And as you've noticed, I haven't given you an answer for the exact, precise division of labor between cerebellum, basal ganglia, and cortex. I have given you some suggestive hints, some guesses. And so how do they work together? Nothing is simpler than its causal structure. The framework for neural causality is neuroanatomy, but I have promised Tony not to get heavily into neuroanatomy, so I won't. Neural systems...
doesn't like details.
Huh?
Tony doesn't like details.
Well, but he's working on very exquisite details in his own work, but maybe he doesn't like details from others.
I love details.
I mean, you know, can you get more detail in the whisker work? I mean, that's so exquisite. I mean, you know.
He doesn't consider that a detail here.
I think we need to abstract away from the detail, but we can't ignore it.
Neural systems work together by being connected, by connective topology, typically in the form of massive fiber bundles whose source to target mappings preserve neighborhood relations. It's a general rule in the brain. There is hardly a system where the connections are scrambled in any way. They are performing coherent transformations from one map to another. The question of how the system or subsystems work together tends to bring to the theory dependence of our conceptual commitments to the fore. The speculative end of this, okay? Which is, as Paul explained so nicely yesterday, the digital computer serves as a massively influential conceptual scaffolding for thinking about brain function. There are nevertheless weighty reasons not to take it too seriously. Here are a few of them, and I wish that Yosha were here, but he's not. 1. Processing speed. I have made my own estimate based on the most recent TMS with transcranial magnetic stimulation. You have an instant interruption of cortical activity. So you can start a process which you have timed with psychophysical methods and physiological methods. You have timed the exact time from somatosensory cortex to V1. There's a study of Braille, I won't get into it. Somatosensory is connected to V1 by multiple relays. You can get from somatosensory cortex to primary visual cortex. You start a process in somatosensory cortex and it ends up in visual cortex that has been timed. Then you start that process and then you see by transcranial stimulation of somatosensory cortex how long you have to wait to interrupt. You put the transcranial on visual cortex and you see how long you have to wait to interrupt that process. Thereby you can measure conduction speed across cortex, including fibers, axons, and synapses. The membrane, the synapses to membrane transformation is the one that takes time. The axon is just splat! You know, the action potential goes down in milliseconds. But what takes time is the membrane potential has to build up, you have to reach threshold, maybe you're not enough at threshold, it just keeps hammering, and finally it goes splat. Once it goes splat, it gets instantly down. So the axon is the Shannon channel of the brain, and it's completely irrelevant. It's just a transmission line. So now you get grand average 135 milliseconds for 250 millimeters. Add 30%, 75 millimeters to the distance because of the curvature of the fiber paths. I did this on the diagrams of the brain. That gives you 325 millimeters summed up, 450 milliseconds for 1 meter, or approximately 2.4 meters per second. That's the conduction time of Cortex. An average conduction time sampled in several places in Cortex with the latest methods that include everything, axons and membrane potentials. The speed of light in vacuum is approximately 300 million meters per second. The electrical transmission in copper wire is approximately 180 million meters per second. That's what electronic components work on. 180 million meters per second. The Cortex works at 2.4 meters per second. Conclusion, neural conduction time, the speed is approximately 75 million times slower than electronic transmission speed. Transistor switching times are measured in the billions or trillions of a second, so they don't add any delay at all. It's only copper wire conduction that determines the speed of electronic transmission. That means that cortical tissue conducts some 7 to 8 orders of magnitude slower. It works 7 to 8 orders of magnitude slower, 75 million times slower than electronic components. Try to build a computer with that kind of components. Nobody in their right mind would even try to do such a stupid thing, okay? The brain cannot, absolutely cannot be like a digital computer. There is no way in hell it could even make a reaching movement. It would take 2 years or something, I don't know. So, forget about digital computers as analogs for the brains. Closely related to transmission speed. I assume there are some computer programmers here. Because of the slow transmission speed in Cortex, the slow conduction speed, because of that, time is always short in the brain. What does a computer programmer do when time is short in his implementation of his algorithm, when he is strapped for time? When time is short, what's the rule for programmers? When time is short, anybody know? When time is short, you don't compute. You use lookup tables. All right? That's what the brain does. It doesn't compute at all, I think. That's my theoretical bias. I don't think it computes at all. It doesn't have time to compute. Even with electronic computers, with transmission speeds of 180 million meters per second, when time is short, you don't compute. You use lookup tables. And the brain uses 2 kinds of radically different lookup tables, but they look the same. One is innately evolved, innate lookup tables. The superior colliculus is an innate lookup table for which eye movement corresponds to which location in visual space. Innate lookup table. The cortex is an acquired lookup table for all the stuff that's stored, okay? The basal ganglia, or probably an acquired lookup table primed by reinforcement learning for what sequences are effective and what sequences are not in behavior, okay? So, when time is short, you don't compute, you use lookup tables. Same thing for the brain. Lookup versus computing. In the brain, time is always short because the sluggish components, they are unbelievably sluggish. So here's a lookup table. Instead of computing, what is this? This is a 12 by 12 multiplication table. Instead of setting up an algorithm to compute 7 times 8, you use a lookup table. Bang, bang, two moves in the computer and you have 56. And this can be done in sophisticated ways, optimally implementation of a Hopfield network. You can set up arrays and you can use lookup tables for optical implementation, and this could be done neurally by divergent and convergent fibers, okay? So now you have what do you have now? Now you have an analog computer for Hopfield, a Hopfield network. And that gets us into analog versus digital. Now, not in the sense of an analog to digital converter. Anything that is analog can be converted to digital with an arbitrary precision. Anything that's digital can be converted to analog with an arbitrary level of precision. That's not the issue. The issue here is analog computers versus digital computers, and they have a huge, huge difference. There was a time when people built analog computers. The Russians lost out in computer technology to the United States because the Russians kept building analog computers. They were unbelievably efficient. They ran their first space shots. They got to the moon with Laika, and they got their first animal up with analog computers. They were dedicated, small, miniaturized analog computers that couldn't be programmed. In an analog computer, the program is built into the hardware. There is no program running on an analog computer. The hardware itself implements the program. Now, that's the kind of computer the brain is. If we're going to talk about computing at all, here is an analog computer for parabolas, a drinking fountain, okay? This is an analog computer. If this reminds anybody of the big arch in St. Louis, it's because both are perfect parabolas. Okay? And if you increase pressure here, you're going to get, if you increase angle and pressure, you can get a complete family of parabolas. You can direct the pressure right up, you can have it go through the roof, and you can have very flat parabolas by changing the angle of the spout of the drinking fountain. Here's another one example. Here's an analog computer. My dear friend Andy Witkin, who died in a scuba accident. He was the smartest of us in my MIT class. Graduate students. There was 6 of us, and Andy was an amazing person, and he died in a scuba diving accident off the coast of California a few years ago. He worked on machine vision, computer graphics and machine vision and computer animation. And for his thesis defense, he built a little toy, which you see here. He built this little toy. It was a little wooden platform, and then he had stainless steel rods going all around the perimeter, meeting at the top, and then, I'll explain the reason for this. Then he had a stainless steel spring, very smooth stainless steel, actually a chrome stainless steel spring, going around this, loosely attached to the rods by small rings, small rings. So this could slide up and down, all around, at all these green places it can slide up and down. And here, it's open. So you can push it up, and these get lengthened, but you still have this. This will compute instantly any conical section by just sliding this stainless steel ring slider up and down. But more than that, the reason he made it for his thesis defense was you can fix, let's say you fix this one and this one, or let's say you fix this one and this one, and you push these up and down. These are going to respond, and you're going to get a smooth curve around that. And he showed by his psychophysical experiments, setting people in front of a computer screen and asking them to judge the surfaces for curves that were identical to the curves he did by pushing these up and down and fixing some of them. And invariably, people chose the same curved surface that he had shown by this simple toy analog computer, this steel spring conformed to when he pushed these up and down after fixing a couple of them. Okay? So he said, look, the visual system is doing something very simple, and I am saying if it's doing it as an analog computer, it needs no more computational machinery than what is computationally equivalent in neural terms to that little toy thing marked up but with steel rods. Here is an analog computer that was found at the bottom of the Aegean Sea next to the island Antikythera, and they discovered from 205 BC an analog computer that computes the position of sun and moon and planets and eclipses. You just turn a knob and it cranks out for the years around which the thing was built. It cranks out the eclipses, the lunar eclipses, by an analog computer, which contained 33 very cleverly interlocked brass cogwheels, okay? Very clever machine. And here is somebody built a modern replica of it. Here is the Greek machine from 205 BC, an analog computer of planetary positions and eclipses. There is no program running on that computer. The hardware implements the solution by cranking the thing forward in time or backwards in time. And here's another one from, this is Paul Churchman's little toy crab robot. It's a toy simulation. The robot has a visual field, a metrical visual field. It has a metrical distorted visual field in arm angular position coordinates. Arm angular position coordinates. You have to transform this to this, and then you simply put them in a 3-layer structure, and you can guide the arm by what the eyes see. And this is implementable by direct analog neural implementation in a structure like the colliculus. And I adapted his idea to a little toy thing for the colliculus. Here you are moving not an arm, but you're taking the information from the eyes and guiding the eyes. And the eyes are simpler than the arm because the eye movements is only azimuth and elevation. Elevation and azimuth. Azimuth and elevation. Two degrees of freedom, or two independent directions of motion. And so you can take the distorted visual space, the distorted in motor coordinates, you can map it onto motor space, and you can get a very simple analog loop that gets the eyes to move to exactly the point where the target appears. Okay. So that's an example of how you might implement this analog-wise. And with that, I have shown my own conceptual biases in approaching the brain. I think it's an analog computer. I think it's forced to be an analog computer by the sluggishness of its components. And when you look at the brain as an analog computer, the solutions you come up with are very, very different than if you think that messages are passed back and forth and things are, here all that is passed back and forth is energy levels, you know, in an analog computer implemented neurally. If you think that there's a program running on your computer, you're going to write a completely different system. So maybe somebody wants to take, I don't know, if you can take up as a project this little one. Implement that in your computer. Anyway, so I had hoped to leave plenty of time for questions, and I hope you have some.